Okay, so welcome to this next video in which we are discussing the different types of mutation. Okay, so we're currently in the process of discussing the central dogma of biology. Specifically, we've got to the translation stage. So we're at the moment discussing the genetic code. Okay, so the basic principle of how you take a piece of mRNA and turn it into a sequence of amino acids is starting with the start codon here. You then work your way along the mRNA looking at combinations of free organic bases. So from the start codon here, you then look at the next free organic bases, then you'll look at the next free organic bases after that. And for each one of these combinations of free organic bases, known as a codon, you will look at the specific sequence of those free organic bases, and you will then find an amino acid that is supposed to be put in for that codon. So for each codon, there is a specific amino acid that is supposed to be put in. Okay, and this will be the amino acid that is attached to the tRNA, which has an anticodon complementary to that codon. Okay, now there are 64 different plausible codons. Okay, however, there's only around 20 proteinaceous amino acids. Okay, now this means that we have far more codons than we have amino acids, which means that there is degeneracy. Okay, so some codons, which are different, will code for the same amino acid. So, for instance, CCA might code for a certain amino acid, and I don't know off the top of my head which specific amino acid that codes for. And there might be another codon which codes for the same amino acid, basically, but the two codons are not the same. Okay, right. So, continuing on the story then, we started with our start codon and we brought in a tRNA which has this complementary anticodon to the start codon AUG, which will always then have attached to the tRNA a methionine amino acid. Okay, then you'll move on to this next codon here, CCA. You'll bring in a tRNA which will have the complementary anticodon here, which will be GGU. Okay, and it will then have a certain amino acid attached to it here. So let's colour this amino acid in orange here. Okay, then what will happen is the ribosome will attach this amino acid, the methionine, to this second amino acid here. So it will form a peptide link between the two, and then what will happen is the ribosome will move along the mRNA. So this codon here, the AUG, will go into the position where these free organic bases here were. Okay, and by the way, this site, so there are 3 sites within the ribosome, okay, which I probably should have mentioned right at the start, but never mind. Okay, there are these 3 separate sites, like so, for codons, basically. Okay, and the way of remembering these 3 sites is this one's called the A site, this one's called the P site, and this one's called the E site, and then it's APE, basically, APE, APE. So what happens is this A site is where the new tRNAs are coming in, which are complementary to the current codon that's there. Okay, and they'll be bringing in an amino acid. Then, in the P site, you have a codon which will have its tRNA still there, and this tRNA will have an amino acid attached. What will then happen is the ribosome will catalyze the attachment of this amino acid onto this amino acid here, and then this amino acid will lose its attachment to the tRNA here. Okay, so to update this picture, what will then happen is the ribosome will move along by 1. This tRNA will now go into the E site. This codon will go with it into the E site. The tRNA will then leave from the E site. That's why it's called the E site. It's for exit. This tRNA here, which now has both of these amino acids attached to it, will go into the P site, and then a new codon will come in. The next codon from along here will come into here, and a new tRNA can come on, and the process can continue. Okay, so let me draw this updated process. Okay, so once again we'll draw our ribosome here. We then have these 3 sites. Okay, we have the A site, the P site, and the E site. Okay, so this is the A site here, this is the P site here, and this is the E site here. The mRNA is coming through here. Okay, so we have now moved the start codon into the E site. Okay, so before the E site we now have this combination that I had here. So the U, the A, the G, and the C are now outside of the ribosome. So here's the U, A, G, C. So they're no longer in the E site. Now the start codon AUG is in the E site. Okay, and its tRNA has now fallen off. Okay, so its tRNA will no longer attach in here. So the tRNA, remember, was U, and then A, and then C. Okay, so let's colour this picture in here. And the tRNA has lost the methionine that was attached to it. So here is the mRNA in green here. Okay, I think we'll colour in the E site in red here. Okay, and this is the site where the tRNAs exit. Okay, and then the mRNA will gradually be moved out. Okay, right. Now we've got the P site in the middle, which is the site where the codon which has a tRNA which currently has the growing polypeptide attached to it sits. Okay, so let's remember which one we had here. We had CCA, so let's put that there. Okay, so we now have CCA here. Okay, and this will have attached to it its tRNA, which had a complementary anticodon, but I haven't got space to put that in. And then it's now got these two amino acids attached to it. Now, obviously, after a while, this will get longer and longer and longer. Okay, so here's our methionine amino acid, and here is our whatever the second amino acid was. And now a new codon will come in here, so let's have GUC here, which will then get another tRNA with another amino acid on it coming in here, so let's put that here. Okay, so this will have the complementary anticodon, so GUC. And then what will happen is this will have an amino acid attached to it, which we'll colour in purple here, and then this dipeptide that we've got here will be transferred onto this one, and then the cycle will continue again. The ribosome will move forward, and it will continue doing this until it reaches a codon in this A site that cannot be matched with an amino acid. Okay, and there are 3 of these codons, and these are known as the stop codons. And these do not have amino acids that correspond to them, basically. Okay, so the 3 stop codons are UAA, also UAG, and UGA. Okay, so these are the 3 stop codons, and at that point you cannot put another amino acid in there, so the ribosome has to stop, and then it will finish the polypeptide. Okay, so that's just a little bit of revision of the central dogma of biology, and that's going to help us when we now discuss mutations. So let's now discuss the different types of mutations. Okay, right. So firstly, let's discuss what the actual definition of a mutation is. So, a mutation is a permanent change to the sequence of organic bases that you have within your DNA. Okay, so in your DNA you have this sequence of organic bases. So let's show this here. So we have A, T, C, G here, C, G again, let's say. So there's a little picture. Okay, this should remain the same always. If you now take one of these pairs, one of these base pairs in the double-stranded piece of DNA, so here we have a base pair, okay, and change it in some way, and that change is permanent within the cell, then that is what's known as a mutation. Okay, so for instance, if we turned this CG base pair either into, for instance, AT, or we could just swap the order of them around. We could turn it into G and then C, so we could swap which strand has the G and which strand has the C, or we could turn it into TA. Those are the 3 different things we could turn it into. That would be a mutation. Okay, now that is a specific type of mutation that I have told you about there. That's what's known as a substitution mutation. So when you just take one base pair and change what base pair it is, that's called a substitution mutation. Okay. However, there are other forms of mutations as well, which will also change the sequence of organic bases within the DNA. You could just delete this base pair completely, basically. Okay. If you delete base pairs from the DNA, that's known as a deletion mutation. Okay. And then the final type of mutation is that you could add base pairs into the DNA. So if I was to, for instance, add in another base pair between this, let's say, let's extend this a little bit more, let's have an A and a T here. If I was to now put in another base pair in between this CG and this AT, let's say here is the CG, and then let's put in a GC, for instance, here, and then we've got our AT. That would also be a mutation. You have changed the sequence of organic bases within the DNA, and that's known as an addition mutation. So these are the fundamental three types of mutation. Either you can substitute one base pair for another base pair, that's known as a substitution mutation, or you can delete base pairs totally from the DNA, that's known as a deletion mutation, or you can add them in, okay, that's known as an addition mutation. So these are all ways of changing the sequence of organic bases within the DNA, and the important thing is that it needs to be a permanent change to the sequence of organic bases within the DNA, which can then be passed on to the daughter cells of the cell. So if the cell was to divide, this mutation will be passed on to its daughter cells. That's the definition of a mutation. It's not just a transient change in the base pairs, which is repaired by DNA repair mechanisms. It's a permanent change in the sequence of organic bases within the DNA. Okay, right. So we now want to discuss the effect of these different types of mutation on the protein, basically, because actually it's more complicated than you would think. These different mutations can have very different effects on the actual protein that you produce, because after all, what's the purpose of DNA? It's to code for the production of proteins. If the mutation doesn't actually affect the cell's production of proteins, then does it matter that you've got the mutation there? Okay, so let's discuss how these different mutations can affect the protein that the gene codes for. So we'll start off with a substitution mutation. Okay, so the first thing is that substitution mutations can be what are known as silent substitution mutations. Okay, so we're now discussing what substitution mutations will actually do to the cell. Okay, so substitution mutations can be silent. Now, what does this mean? Well, this means that it has absolutely no effect on the cell, basically. Now, how can this be? Well, one potential reason is that the mutation has occurred in a non-essential piece of DNA. Okay, so you have a huge amount of DNA within the nuclei of your cells. Okay, not all of it actually is used to produce proteins. Okay, so if I draw this, let's say this is an extremely long piece of double-stranded DNA. There will be dotted around here genes. Okay, so these portions that I'm outlining here, these are going to represent genes. Let's colour them in. So here's a gene in green here, here's another gene here, and then there's portions in the middle which are not actually used to produce protein. Okay, this will be a non-essential piece of DNA. If you change a base pair here, it's unlikely to have any effect, basically. That's what I mean by a non-essential piece of DNA. Okay, so that's a mechanism by which a substitution mutation can be silent, that it occurs within a piece of non-essential DNA and therefore doesn't really affect the cell's production of proteins at all. However, substitution mutations can also be silent even if they occur in genes, and this is cleverer, this is more subtle. Okay, so how can that be? So let's draw out a piece of our gene here. Okay, so here, let's say this is our gene here, and we'll have some organic bases here. So let's have GACT here, and then we'll have the complementary sequence on the other strand, CT GA. Okay, right. So let's now say that we are going to substitute one of these organic base pairs here for something else. Okay, how can that be? How can it be that that is not going to actually affect the protein in any way? Well, one suggestion is that it could be within the introns, so that's a good idea. We'd count that also as non-essential DNA. So if it occurs within the introns, then yes, it's not actually going to be put into the mRNA, and therefore it's not actually, well, it's not going to be put into the mature mRNA, it'll be put into the pre-mRNA, but it's not actually going to be translated into protein. So that's one way that it can be a silent mutation. But let's say that this is actually occurring within our exons. Okay, so that means that when we finally do have our piece of mRNA here, so let's say this is our mature mRNA, you are actually going to end up changing one of the organic bases in this mature mRNA to something else. Okay, now why is it that that is not going to affect the protein? Well, another trivial suggestion is that it could be upstream of the start codon or downstream of the stop codon, in which case, yes, of course, it won't affect the protein. But let's say it actually is in between the start and the stop codon, and it is going to be translated. So let's put this in here. So here's the start codon, and then we've got, let's say, the second codon is a CCA here, and we're now going to do a substitution mutation, and we're going to change this A into, let's say, a G instead. Okay? Now, I don't actually know what CCA and CCG actually code for, but let's use this to highlight the principle. This might not actually be a silent mutation, but let's suppose it is going to be a silent mutation. How could it be a silent mutation? Well, what if the codon CCA coded for the exact same amino acid as CCG? Remember, we have 64 different codons, okay? Not all of them can code for distinct amino acids. Some of them have to code for the same amino acid. So if CCA coded for the same amino acid as CCG, then the actual polypeptide that you produce will be exactly the same. It will have exactly the same amino acids, because the amino acid we'll put in for the second codon won't change simply because we have substituted one organic base for another organic base. Okay, so that's how this substitution mutation could potentially be silent, that it's not actually going to change the protein at all, even though it's within a portion that actually is going to be translated. Okay, so if these two both code for the same amino acid, okay, then that would lead to this mutation being a silent mutation. And as I say, I don't actually know which amino acid CCA and CCG code for. Okay, so I don't know if they're the same. So this might not be a silent mutation, but just to show the principle, there you are. Okay, right. Now, let's suppose that this mutation CCA to CCG, which is this substitution mutation, let's suppose now that CCA and CCG don't code for the same amino acid. Okay, that's more serious then. That would cause what's known as a missense substitution mutation. Okay, so now let's turn our attention to missense substitution mutations. So this is where you change one of the organic bases in one of the codons within the mRNA, okay, that's actually going to be translated, and the new codon that you produce does not code for the same amino acid. So that means in your final polypeptide, okay, so let's say our polypeptide is here, one of the amino acids is going to be changed. You're going to take one amino acid, let's say it's this one here, in turquoise, and you're going to change which amino acid it is. Okay, that might change the properties of the protein, okay, and that then is called a missense mutation. Okay, so when you change one amino acid within the polypeptide because you've changed one organic base within the mRNA here, that's called a missense mutation. Right, next up, you have what's known as a nonsense mutation that can occur due to substitution. So it's a nonsense substitution mutation. Okay, now what happens here, in this case, what you do is you mutate one of these organic bases. okay, in a codon, and it needs to change the codon from being a codon which codes for an amino acid to being a codon which codes for a stop codon. Okay, so let's put this here. So let's say we've got AUG here, okay, and this is our mRNA here. Then we've got the CCA portion, and then let's say after that we had, what should we have? Let's say we have UAU. Okay, so let's now say that we make a substitution mutation in our DNA that then results in the change in this organic base here. Okay, and let's say we change it from a U to an A. Okay, so let me just colour this mRNA in green. So this is our mature piece of mRNA. So previously UAU had some amino acid that it coded for. Now we've changed it to UAA, which, remember, is one of these stop codons. Okay, so we have inserted in this stop codon, and we've inserted it in as codon number 3. Okay, that means that we're just going to attach methionine to whatever amino acid is coded for by CCA, and then we're going to stop. Okay, we should be going on for, you know, hundreds and hundreds of organic bases yet. But now we've stopped translation really early. Okay, so this is what's known as a premature stop codon, and it may be utterly disastrous for the synthesis of the protein. For instance, here it would take the synthesis of potentially a massive great protein down to joining 2 amino acids together. Okay, so this is called a premature stop codon. So in a nonsense mutation, what you do is you change one organic base, which changes the codon from coding for an amino acid to being a stop codon. That leads to the premature halting of the translational process, and therefore your protein is cut off early, basically. And depending on clearly how far forward the premature stop codon is, that will have differing levels of severity for the function of the protein. Okay, so those are all the different types of substitution mutations. Let's now turn our attention to addition and deletion mutations. Now, addition and deletion mutations are generally more severe than mutations which are just substitutions, and let me explain why. So we'll start with addition. Okay, so if we are going to add in an organic base, okay, so we're going to add in a base pair of organic bases, so this is addition mutations, then firstly, addition mutations can be silent, okay, and this will occur if they occur in non-essential DNA, okay, so if they occur in the portions in between genes, then it won't affect protein production. Okay, or if they occur within the introns of a gene, then again it won't affect the protein production. However, let's assume that it occurs within the exons. Then the overall result of that is going to be that we are going to add in an extra organic base into the mRNA. So let's say this was our original mRNA here. So here's our AUG-CCA. Okay, so this is our mature mRNA here. And let's now say, well, actually, let's put another one in here. Let's then have ACG there. Okay, so let's say this was our mRNA before the addition mutation, and now let's say we're going to add in a new organic base, and we'll add it in between C and A. Okay, so let's now put in a G there. Okay, so we'll now put a G in here, and now here's that A there. Then we've got the A, the C, and the G. Now, why is this so severe? Well, let's think about how we would have translated this protein up here. So we'll start at the start codon here. We'll put in an amino acid for that start codon. We'll then move on to CCA. We'll find the amino acid for CCA. We'll then move on to ACG. We'll put in the amino acid for ACG. Now, let's look at the mutant one down here. We'll start with AUG, we'll put in the amino acid for AUG. We'll then move on to CCG, we'll put in an amino acid for CCG. We'll then move on to AAC. Oh, wait a second, now you see it's having more consequences because we are now not reading the ACG properly anymore. It's not just affected this codon here, as was the case in the substitution mutations. You only really affected one codon. Now it's affecting all of the codons because ACG isn't going to be read properly because you've shifted the reading frame along, okay? You've moved it from reading in this frame here, ACG, and then onwards after that. You've moved all of the frames along by one, and the codons now are going to be completely different. So the sequence of amino acids that you are actually going to put into your protein is now going to be completely different, basically, after this addition mutation. Okay, so this concept that we are moving all of the frames along and therefore looking at completely different codons from the point of the addition mutation onwards, basically, this is called frame shifting. Okay, and these can be very severe. You can get a totally different protein being produced, okay, because you've totally shifted the frames of the codons that you're looking at, and therefore the sequence of codons is completely different now. Okay, right, and similar frame shifting can occur if you have a deletion mutation. Now you might be able to spot a way around this. If you were to add in free organic bases, okay, so if you were to do an addition of more than one organic base, if you were to add in a free organic bases or a multiple free organic bases, then it would not produce the frame shifting like this, okay. It's if you put in a number of organic bases that is not a multiple of free that it then causes this frame shifting, which then means that the sequence of codons is completely changed, the sequence of amino acids in the polypeptide is going to be completely changed. Okay, now the same principle holds true if we delete one. So imagine we delete this A here, then what you will move the frame now to is you'll read CCA. Okay, that's brilliant. That one codon was unaffected. But then you'll go on to CG and whatever follows that. And then after that, you'll have shifted one by one again. You'll have shifted by one in the opposite direction, but it's still a shift by one, basically. And again, you'll have a completely different sequence of codons, which will then code for a completely different sequence of amino acids. So that now concludes our discussion of the different types of mutation.